In this video, we're going to be talking about the skeletal muscle fiber anatomy and sarcomere anatomy in more detail. The sarcomere is really part of the contractile unit of a skeletal muscle, so ultimately its function there then is to help produce contraction. So let's kind of refresh some of the skeletal muscle fiber anatomy. So the skeletal muscle fiber is going to be the individual cell. So the individual cell is going to be surrounded by the sarcolemma. So that's going to be the overall membrane that surrounds them. Really, the membrane portion should be this, that it'd be better if it pointed at this inner portion here. The overall structure of the muscle fiber, note that it has multiple nuclei in a single cell, has lots of mitochondria. The individual contractile proteins are organized into units known as myofibrils. This is where you're going to find the sarcomeres, with the light and dark bands that ultimately are going to regulate muscle contraction. And then the sarcoplasm is going to be the portion of the cell that contains the cytoplasm of the cell. All right, so let's look at a longitudinal view of the myofibril. The myofibrils are going to be rod-like contractile elements. They make up most of the muscle cell volume. They're composed of sarcomeres, which are arranged end to end. So this is one sarcomere that you see there. The sarcomeres and the myofibrils are going to appear banded, and bands of adjacent myofibrils are going to be aligned. That is why you have that striated appearance in skeletal muscle. The overall sarcomeres are going to be made up of thin filaments. So the thin filaments are the actin fibers, and then you're also going to have thick filaments, which are the myosin proteins. The myofibril will be wrapped around, or wrapping around the myofibril will be the smooth endoplasmic reticulum. What we do is call it the sarcoplasmic reticulum in muscle cells. Its primary function is going to be to act as a reservoir to hold calcium ions, which we'll see later play an important role in muscle cell contraction. All right, so let's look at a single sarcomere structure now. So first of all, we have light and dark bands. The light bands are known as the lighter I bands, and the darker bands are the darker A bands. Striations are going to be evident along the entire length of the myofibril. If the muscle is intact, this alignment is nearly perfect, such that you have those dark and light regions. Each A band is going to have a midsection that it has slightly lighter, that is known as the H zone. The H zone is going to be bisected in the middle by the M line. So the M line is going to bisect the H zone. Meanwhile, the I band has a midline interruption. This is going to be a darker line in the lighter region that's known as the Z disc or the Z line. So the sarcomere is going to go from one Z line to the next Z line, and that is considered the functional unit of a skeletal muscle fiber. The central thick filaments of myosin extend the entire length of the A band. They are connected in the middle of the sarcomere by the M line, as I mentioned. If you look at the thick filaments, they are going to be made up of your myosin heads that come off from them. while the thin portion are going to be made up of the um, thin filaments. So let's take a look at the sarcomere structure in a little bit more detail, looking at the individual units that make up the thick and the thin filaments. So first of all, the thick fibers are going to be made up of myosin. The myosin molecule is going to consist of 6 polypeptide chains, 2 heavy chains and 4 light chains. The heavy chains twist together to form the myosin's rod-like tail, and each heavy chain ends in a globular head that is attached to the tail via a flexible hinge. So you can see the heads right here, the tail of the myosin molecule extending back, and then you have your flexible hinge region there. The heads are going to contain actin binding sites, so these are going to be the sites that are going to interact with the light chain. Could be thought of as the working end of them. So during contraction, they link the thick and the thin filaments together, forming cross bridges and swivel around their point of attachment, which act as a motor to generate force. Myosin itself will be splitting ATP, so it acts as an ATPase and uses that energy that is released during this reaction to drive the movement of the muscle. So each filament is going to contain about 300 myosin molecules bundled together, with their tail forming the central part of the thick filament. All right, the thin filaments are going to be composed chiefly of the protein actin. So actin filaments consist of kidney-shaped polypeptide subunits. These are called globular actin or G-actin. Each G-actin has a myosin binding site to which the myosin heads attach during connecting. The G-actin subunits will polymerize into long filaments called filamentous or F-actin. And then you have 2 intertwined actin filaments resembling a twisted double strand of pearls that form the backbone of each filament. Thin filaments also contain important regulatory proteins. So the first one is tropomyosin. Tropomyosin is a rod-shaped protein. It is going to spiral around the actin core and help to stiffen and stabilize it. The subsequent tropomyosin molecules are going to be arranged end-to-end along the actin filaments. In a relaxed muscle tissue, they're going to actually block the myosin binding site that I mentioned earlier. So that dark green structure there, that is your myosin binding site that's blocked by the tropomyosin that we see here. The other regulatory protein is going to be the troponin. Troponin is a globular protein with 3 polypeptide subunits. One subunit attaches troponin to actin, another binds the tropomyosin and helps position it on the actin, and the third subunit is going to bind to calcium ions. We'll see how those are important later. All right, another feature of the sarcomere is that it is going to be covered by the sarcoplasmic reticulum and a structure known as a T-tubule. T-tubule is a protrusion of the sarcolemma that forms an elongated tube. The sarcolemma you recall is the plasma membrane, so it's going to extend down over the myofibrils and encircle them along this route. The T-tubules then are these extensions. The lumen of the T-tubule then is going to be continuous with the extracellular space, so you can see this is open to the outside of the cell. This permits the conduction of electrical impulses very rapidly, and it's going to, with portions of the sarcoplasmic reticulum, act as a um, as a triad. So you have this terminal cisternae of the sarcoplasmic reticulum. Then you have the terminal or the T-tubule, and then another terminal cisternae that make up the triad. These help communicate to the sarcomere when contraction is supposed to take place, because the signal is going to come from outside the cell, and this allows it to interact with the sarcoplasmic reticulum, and ensuring that the myofibril contracts in sync with one another, because they're going to be continuing on as it continues down. This keeps everything in contact, so all the myofibrils contract. All right, we have a model of muscle contraction that is known as the sliding filament model of contraction. During the sliding filament model of contraction, the I band is going to shorten. As the I band shortens, the distance between successive Z discs shortens, and as the filament slides centrally, the Z discs to which they attach are pulled towards the M line. So again, you can see what's happening here as the muscle contracts. The I bands shorten, the distance between successive Z discs shortens as they are pulled towards the M line. The H zones are going to shorten and then disappear. The length of the A bands is going to remain the same, and the distance between the A band is going to shorten. All right, at the neuromuscular junction, this is where you're going to get the signal for contraction to actually take place. So you have a motor neuron with the axon terminal synapsing with the individual muscle fibers. The motor end plate is the location of the acetylcholine receptors in the muscle fiber sarcolemma. Acetylcholine is the molecule that is going to trigger the contraction to occur. All right, so when the action potential reaches the axon terminal, vesicles containing acetylcholine will release acetylcholine into the synaptic space. So we can see the action potential going down to the terminal of the axon. The vesicles that contain the acetylcholine get released into the synaptic cleft. The motor end plate is going to be the portion of the muscle that is going to synapse with the axon terminal, and that is going to be where you have an acetylcholine receptor, which is a gated ion channel present. So acetylcholine binds to the acetylcholine receptor. Binding of acetylcholine to the receptor is going to open that receptor. It's a channel that allows sodium to go in. So as sodium goes in, it's going to result in a local depolarization at the end plate. This is known as the end plate potential. The end plate potential is going to trigger an action of potential across adjacent sarcolemma, and so that's what's going to trigger overall contraction. So how does the action potential lead to contraction? Well, it has to do with the function of the actin, myosin, troponin, and tropomyosin. So you have your thin filaments that are currently binding to troponin and tropomyosin. The troponin is going to hold the tropomyosin such that the myosin binding site is being blocked. The troponin can bind to calcium, tropomyosin, and actin When calcium is absent, then the troponin is going to keep the tropomyosin in place and blocking the myosin binding sites. So let's look at what happens as the action potential moves into the muscle cell. So action potential is going to move as voltage-gated sodium channels open. So we start with that happening. The positive charge moves along the membrane. So the action potential then moves down to the T tubule. The membrane depolarization that is happening here is going to trigger calcium release from the sarcoplasmic reticulum. This in turn leads to extracellular calcium ion increasing in concentration. The calcium ions then are going to be bound by the troponin. Binding by troponin is going to move the tropomyosin out so that the myosin binding site is unblocked. Now you can have actin-myosin cross-bridging and ATP hydrolysis occurring subsequently, leading to contraction. So a cross bridge is going to form between the actin and the myosin head, triggering contraction, and as long as the calcium ions remain in the sarcoplasm, they are going to, and bound to the troponin, and as long as ATP is available, the muscle fiber will continue to shorten. Now, when the muscle relaxes, what is going to happen is that your calcium ions are going to be removed from the synaptic cleft. So all of these little red dots get removed from here. Going to be the calcium ions are going to return to the sarcoplasmic reticulum as the membrane repolarizes. So we can see the calcium re-entering the sarcoplasmic reticulum. That decreases the overall concentration of calcium. You can see that calcium no longer is binding to the troponin, and therefore the tropomyosin is blocking the myosin binding sites on the actin, and therefore you don't have actin-myosin cross-bridging, and therefore the muscle will lengthen and relax. All right, that does it for the microanatomy of the muscle relaxation. We'll talk more about the action potential and the muscle twitch in the next video.